طيب. Now, in this lecture, we are going to study the next experiment that happened after Griff, okay, and done by a scientist called Oswald Avery. Avery, as you see his picture here, he did a unique experiment based on the uh, uh, Griff experiment. What he did is the following. Now, every team spent 10 years trying to figure out what was this transforming principle. Okay, شو هذا الشيء, that thing that moved from the heat killed S bacteria to the R bacteria, and change the R bacteria to become stronger and to have shell. In another word, this R bacteria became S bacteria by taking something from the heat killed S bacteria. Okay, now, he did something unique. He decided to prepare an extract from the S bacteria. Now, you can ask me, what is extract? What is an extract? When you prepare an orange juice or any kind of juice that has debris, okay, and you purify this juice from the debris. لما بنحضر أي نوع من أنواع العصير وبنتخلص من الأشياء الغير ذائبة وبنحصل على عصير إيش؟ على عصير صافي. That's an extract. The same thing he did. He collected a lot of S bacteria, and he break down this bacteria, and he extracted the soluble things from this bacteria. استخرج منها الأشياء الذائبة بالماء. And he had an extract in a test tube. تخيلوا التست tube has extract. شو سوى? And he had a petri dish, okay, and he mixed the R bacteria with this extract from the S bacteria, and he let the bacteria to grow on the petri dish. حط البكتيريا على وسط غذائي وخلطها مع ايش؟ مع ال S extract. يعني حط R بكتيريا وخلطها مع ال S extract وتركها. لاحظ انه what؟ انه the new bacteria that grow has the S bacteria property, which is having a shell that protects the bacteria. But what is this thing inside this extract that changed the R bacteria to become what? To become S bacteria. He started to study this extract. He was smart. He took this extract and he did 3 different tests. The first one is a qualitative test, chemical analysis, and enzyme treatment. Now after he did this, the qualitative test showed that the DNA was present in this extract. لقى انه في DNA موجود في هذا الاكستراكت اللي غير الار بكتيريا وحولها لاس بكتيريا. بالchemical test showed that chemical makeup matched that of the DNA. اجى فحص شو قال شو في chemicals موجوده بهذا التست تيوب بهاي الانبوبه هاي شوفي فوجد انه the chemical component of this test tube matched with the DNA component. هذا اللي بتشوفوه هون بالتيبل هذا. Don't worry, I'm not going to ask you about these number of details, but just know it that when he did a chemical test, the chemical test showed and matched the chemical makeup of the DNA. The second thing, the third thing, he did an enzyme test, okay? The enzyme test showed only DNA degrading enzymes stopping transforming. لما كان يمسك التست تيوب ويحط عليه enzymes, and those enzymes can break down the DNA. Hello? This enzyme لما يكسر DNA كان يمسك هذا الاكستراكت بعد ما يكسر DNA and put it on the R bacteria, mix it with the R bacteria and let it grow, the R bacteria stay R bacteria. In another way, لما كسر DNA اللي موجود بالاكستراكت وعمل ميكسينج للاكستراكت هذا اللي فيه دي ان اي مكسره خربانه، عملها ميكس مع الار بكتيريا، ما طلع عنده اس بكتيريا، يعني ما تحولت الار بكتيريا. فمعناها هو صار almost there. He said almost. I'm pretty sure that DNA is the transforming principle. DNA is the genetic material. He said this. Okay. Now the third experiment after Griff experiment, every experiment we're gonna study Hershey and Chase experiment. Those two scientists did a very sophisticated somehow experiment. We're gonna watch a video about it and we're gonna study it. Next video.